Hello, and welcome to this week's episode of I Came for Science, a show about sex and reproduction and inclusion in STEM. I'm your host, Laura Surratt. I'm a professor of biology, and I've been studying animal sex for the past 30 years. Please join me as we normalize talking about sex and what we know about it, as well as the journeys of the scientists who study it.
And today we're going to switch things up a little bit. You've been hearing primarily from Laura, but today we're actually going to interview Laura as the focus of this episode. As a reminder, my name is Natalia. I was Laura's student and I graduated in 2014 and we've remained in a close mentor friendship ever since. So that's just a little bit of an introduction. So Laura, why don't you start us off by telling us about yourself?
Sure. Thanks so much, Nat. This is really fun to be doing. I can't believe we're here today with you interviewing me and
I know,
us having this discussion. It's great.
It's crazy.
So let's see. I am currently a professor and chair of the biology department at the College of Worcester. And my area of research is on animal reproduction. I basically love studying animal sex and am very interested in all things related to it, from the physiology, the molecular biology, the evolutionary biology, the behavioral ecology, all aspects of it. And most of our research right now is on mosquito sex, but we also study human sex from the perspective of using evolutionary approaches to address human infertility problems. So that's me. And then I teach and do mentored research. So I teach animal behavior, I teach evolution related classes, ecology related classes, and an invertebrate biology class, natural history of the invertebrates. I also, together with my students, started a class called Canine Coach, where we work with the local dog shelter to help recent adopters with the transition of having a new dog in their home. So that's it. And I do some of my research in Costa Rica on mosquito ecology there, just because I love working in different countries and meeting researchers from different countries. So that's been a really fun part of my job as well. And then the final part is our college has every single student do a thesis. And so a big part of my work is working side by side with students on research, like we did. We started off together and then you went off and worked with other people, which was great.
That's right. Can you tell me a little bit about what got you interested in animal behavior and insect sex and human sex and all of those things?
Yeah, absolutely. So maybe like a lot of kids, I just loved animals growing up, and we had dogs growing up. I started working at a nature center and then at a zoo, where mostly my job was to scoop the poop of a lot of different animals, from like all different exotic animals. I scooped the poop
Character
of.
building
So
stuff.
yeah, absolutely. And so there was that. At about the age of 11, my grandmother told me that she knew someone who studied animals, and her name was Jean Altman, is Jean Altman. And she put us in touch with each other. And so we started writing back and forth. And I was like, wow, this is a real job. So it was at 11 that I realized that I could have a job. Somebody would literally pay me to watch animal behavior, my very favorite thing in the world. And that blew my mind. And so I kind of just kept that on my mind for the next few years and got interested in a number of other things and went to college, and was fortunate that my very first semester I took a primatology class and we read a paper by someone named Jean Altman. And I was like, wait a second, is this Jean Altman? I was like, oh my God, I've been writing with someone who's famous. And she is really famous. She's done so many incredible contributions to the field of primatology and animal behavior and ecology and reproduction. And I had no idea if she was sort of famous in the field. I just knew her as this friend of my grandmother's. So that's how I got into it. I was really lucky when I went to University of Michigan. There were 5 world famous primatologists there at the time. And I just went from one primate behavior class to another and loved it all the time. And then in the summers, I got jobs working as a research assistant to collect data on primate behavior. So I was very much in the mindset, I'm going to be a primatologist. I'm going to work on primate conservation and go into that field. But that's not what happened.
Yeah, how did you get from primates to mosquitoes to
humans? Yeah, I'm not sure how much time you have here, but I'd say there are a few different things that converged. So one was that I wanted to take biology at University of Michigan, and at University of Michigan they would not allow you to take biology unless you were willing to dissect. You had to dissect animals as part of the intro bio curriculum, which is reasonable but not something I was willing to do. I had had a really bad experience in middle school with animals not being treated well in anticipation of them being dissected, and I hated it, and I vowed never to do that again and didn't want anything to do with it. So I said, I will watch, but I don't want you to get animals for me. I will just watch the dissections. And the University of Michigan said, no, you can't take biology then. And so I never took biology as an undergrad. And instead, I majored in anthropology and mathematics and was planning to go to grad school to study primatology, which is interesting because studying primate behavior is in three different fields, really. You could be a biologist studying primate behavior, you could be a psychologist studying primate behavior, or an anthropologist studying primate behavior. So
Wow,
those are
that's
sort of
interesting.
the three academic disciplines, at least, that look at primate behavior. So I actually took 3 years off after undergraduate. I took my very last class at University of Michigan was with a professor named Bill Stapp, who is a leader in environmental education. And I put on the brakes and I said, oh my gosh, I think I love this instead. So I said, I'm not going to grad school. I'm going to go teach environmental ed. So for a few years, I taught environmental education in Connecticut, and then decided I was really being called back. I was just needed to learn more. I was so curious and loved the academic space of just having research and questions and challenges to your ideas all the time. And I missed that. And so I applied to all anthropology programs and one biology program at University of Florida. And that's ultimately the one I decided to go to work on primate conservation with a professor named Colin Chapman. I wanted to work, I knew I loved reproductive behavior because I'd studied it as an undergrad when I was a research assistant, and I wanted to do conservation. And to me, those two things meshed logically. Like, it made sense. Like, they have to reproduce to survive, right? There must be something. But my committee encouraged me to study ecology instead, which is about how organisms interact with other organisms or abiotic, non-living things in their environment. And so I wrote a whole proposal to do that, but I didn't want to do it. So it was a good proposal, but it wasn't something I actually wanted to study. And so I decided that must mean I have to leave graduate school. And I already had a huge imposter syndrome because I'd literally never taken biology, and I was in a biology PhD program. And so it wasn't just that I thought I knew the least in the room. I literally did know the least in the room, and there was a lot for me to learn.
Wow.
And I just thought, not for me. This is just playing into my insecurities too much. I'm miserable here. And so I started telling people that I was leaving grad school after a year. And one person on my committee, Jane Brockman, said, you love reproductive behavior, right? And I said, yeah, I really love it. And she said, well, there's these great damselflies, which are a relative of dragonflies, that live in the retention ponds, these like wastewater ponds essentially behind our building. There's a really cool system to study. Why don't you study them? And I was like, no, 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 I'm a primatologist. That's what I'm doing with my life. She said, just do this for your master's, study them for your master's. You can go back to primatology later. And so she really pulled me back into grad school, which I'm so grateful for. And then I studied the damselflies. And then from there, I was kind of like, oh my God, there's a million questions to answer with reproduction of insects. And with primatology, there's relatively not that many species of primates and a lot of people who are interested in studying them. And it's almost the opposite with insects. There are so many species of insects, and definitely not enough people to study all of them and all aspects of their behavior. And so it just was like a field day for me to be like, oh my God, there's all these species that we don't know anything about their reproduction. So that's how I got into insects.
Yeah, access alone must have been very important at that stage because it's rather than going to another
I was supposed to go
country
to
to study
Uganda.
primates,
Yeah. Yeah.
you can go
Yeah.
behind your building to the retention pond and look
Right.
at
Yeah.
damselflies. So,
Yeah.
well, that's really interesting. I think one thing that I've always taken away from talking to you is that it's okay to not stick to something if it's not working out and you still love primates, but like look at all the other opportunities that have opened up because you were like, you know what? Okay, damselflies to get through my masters, let's just do it. And then that
Mm-hmm.
opened up this whole other
Whole other
window for you.
world. Yeah.
Yeah, I
Yeah.
love that.
Door I didn't even know was there. I'm so glad you took that away. The other thing I hope that people in my life take away from my experience is that it's okay to know nothing. and build up your knowledge. Like it's okay to be a beginner again. So just because you're 20, 25, 30, 35, 40, 50, whatever it is, it's okay to start from scratch. And that your previous experience is going to bring a new perspective into that field. So, me coming from primatology into entomology, I don't know anyone else who's done that. I'm sure there's people in the world, but it's not a common switch. So, I had all this primatology knowledge that I was now bringing to the field of entomology and could offer new perspectives. And I feel like that's something that's been really exciting about my career, that people have really valued the fact that I'm bringing in information that they didn't have, even though I don't know anything about this new field I'm coming into. So just realizing that it's okay to start from scratch as long as you're willing to learn.
Yeah, speaking of starting from scratch, so that gets you to grad school where you're studying damselflies,
Mhm.
but then your career took a different turn. Do you want to talk
Yeah.
about that a little bit?
Yeah, and the first part of that turn was when I went from my master's to my PhD. So I was studying damselflies. They're so full of wonderful sexual behavior and sexual positions and body parts and all sorts of things that we could talk in depth about. But the ones I was studying at least didn't have an applied problem. And I really wanted my research to have applied implications, like solving a problem in the world. And I thought that would be in conservation. But I started working for on my PhD on this grasshopper group that was of conservation concern, and I was looking at how they had evolved through sexual selection, so how their body parts had evolved. And it turned out I could not get these grasshoppers to mate for my life. Like everyone's like, grasshoppers, they mate all the time. I see them mating all the time. And when I want to watch them and see how the mechanics work, they would not mate for me. And so after a year, it became clear that I had to switch projects. I kept trying and trying, and we would joke that we were playing romantic music for them, all sorts of things, dimming the lights, but it wasn't working out. And so I pretty quickly needed to turn
They're
around and
shy,
find.
Laura. They didn't
I know that, and I
they
was
didn't like being
voyeuristic.
watched.
Yes. So my PhD advisor Jane had suggested that I study a beetle that was a pest species of citrus in Florida. It was not only of citrus, it also attacked horticultural plants, and it was making the plants that it attacked vulnerable to disease and dying. And if you threaten citrus in Florida, you're sort of enemy number one down there. And so the US Department of Agriculture was working on them, and they wanted someone to study their reproductive biology. And she thought that would be a good fit for me. And it really took a village to convince me to work on pest management, like a village of my friends and colleagues, because I just said, I don't want to kill pests. I mean, I don't want to kill insects, right? I love them. I want to conserve animals. That's what I was supposed to do, conservation. And so what some of my colleagues suggested was, like other grad students said, well, you can do conservation through pest management. If you're on the inside of that, you can work on species specific approaches to controlling particular insects so that we're not using these pesticides that affect lots of different species. And so that's really what my career has become, is looking at species specific approaches to controlling reproduction of species that we consider pests. And for my PhD, I worked on a beetle species, and the females mated with tons of males all the time they were mating. And the question was, what were the predictors of whose sperm the female would use? So in insects, females can store sperm. It's not just insects, even humans, you should know that too. That sperm can stay alive in the female reproductive tract. So I was studying when females mate with multiple males, what predicts which male sperm she used. And so that was when I was working with the pest species. And then I kind of continued along that curve. I had not learned any molecular biology or biochemistry during my graduate program, very, very, very minimal amounts of that. And I just thought, if you're going to be a well-rounded biologist, you really need to have all aspects of biology. And so for my postdoc, which is what people often do, like a research program people do after their PhD and before they start an academic position or industry position, that I would learn biochemistry and molecular biology. And one of the researchers at USDA suggested the lab of Mariana Wolfner, who we talked to in an earlier episode,
Mhm.
and said she works on exactly the questions you do, but just on the molecular and biochemical level. And the questions I was interested in that point were, how do different individuals influence each other's reproductive success? And she studied it in terms of these proteins that males transfer to females during mating. They're called seminal fluid proteins. So I started studying these seminal fluid proteins, but in a fruit fly, Drosophila melanogaster, who also, that species is also not a pest or of conservation concern. And so pretty quickly after that, we started a collaboration, Mariana, myself, and Laura Harrington, who was in at Cornell Department of Entomology, and she studied Aedes aegypti, which is a species of mosquito that transmits lots of different pathogens, including the ones that cause dengue, yellow fever, Zika, chikungunya, and other diseases as well. And we started studying the seminal fluid proteins in them. So that's what we've continued in my lab. So that's how I got to mosquitoes and what I'm studying now with mosquitoes.
Wow. Yeah, that is fascinating. You're talking about the beetles and how the females decided which sperm to use. And my question is, what mechanism do they use to even sort through their library of sperm?
I love that. I've never heard that term before, library of sperm. It's an excellent question. And so I think the way I phrased it, or the way I should have phrased it, is that we were looking at the predictors of what sperm were used. And I kind of try to be careful of not saying that females choose the sperm from different males, but there is something we call cryptic female choice. And that's this idea that females or female behavior or physiological processes can influence male reproductive success of the different males she's already mated with. So again, females can store sperm from different males. And in insects, they're stored in a structure. Most insects have something called a spermatheca, which is like this hardened structure that sperm can live in, sometimes for days, months, even years in some species. And in some species, like the mosquitoes I study and in fruit flies, there's multiple sperm storage organs. So it's tempting to be like, okay, well, maybe she puts one male sperm in one of those organs and the other male sperm in another organ. And we don't see evidence of that. But we do see that the female structures, like there's tubes that lead to those structures, that they're innervated. So there's a lot of neurons connecting to them, and there's muscles and there's valves in some species. So there seems to be mechanisms by which females could selectively release certain sperm. But beyond that, there's other things females can do. So when a female mates with a male, we know in several different species that females can eject the sperm. So the male's ejaculated and she can squirt that sperm right out at different times. And
so if a better male sperm comes along, she can like empty the bank, refill it.
Usually it's right after the first mating, but that point that you made is whether she mates again with another male is another form of cryptic female choice. So she's mated with one male, and maybe a male that has some qualities that make him better in some way comes along. She can decide whether to mate with him or not, because in most insect species, there's what's called last male sperm precedence.
The last male she mates with gets preference?
Yeah, so it's sort of like last in, first out, basically. So the last male sperm, the last male she's mated with, his sperm are stored maybe closest to the exit of the spermatheca. And so those are the ones that'll come out first and fertilize the eggs in some species. Okay. And so, by mating up, sort of like each male she mates with is maybe better than her previous mates, those are the sperm she'll be more likely to.
That is so fascinating. So I wonder if she gets more selective each time.
Right. Yeah. And you see that in some species that unmated females are less selective than already mated females. So I just want to go back to the damselflies for a second, because just like females might have mechanisms to release sperm selectively, and they have the last male sperm precedence in some species, males also of some species have evolved mechanisms to get the sperm of other males out of female sperm storage. Starting with the beetles that I told you about. So the males in that species, there seems to be cryptic female choice going on. The matings last for up to 16 hours. And as a grad student, I stayed there and I watched every single freaking thing they did for 16 hours. And one thing I noticed that people hadn't noticed in the species before is that the males would tap the females with their antennae and stroke the females' underside with their legs during mating. So I counted every time they did that during those long matings. And it turns out if you mate a female with two different males, it's not the last male whose sperm is used. It's the male who has stroked the most. Yeah, with his
Oh,
antennae. It's
the one
great.
giving her the best massage.
Yeah, exactly. I mean, we call it copulatory courtship is the name in the field, right? And that somehow it's an indicator maybe of male endurance or his energy stores or something that the female is able to exert this preference and use his sperm. But there's more. Damselflies are just a classic example of males kind of manipulating the system of sperm storage. So male damselflies have amazing penises. They, depending on the species, they have spines, they have scoops, they have curlicues, and those parts of their penises can get into the female sperm storage organ and scoop out the sperm of previous males. And there's other ways they can sort of stimulate the female to eject the stored sperm. So it's amazing.
Yeah, so how did you get from studying this fascinating mechanism in insects to studying it in humans?
Yeah. I never thought I would be studying humans. It's so funny because, I came from this primatology background and I've always wanted to go back and study monkeys, and I've done a little of that, but humans were not on my radar as a study species at all. So it really was a student who motivated it. It was awesome. Jamie Innes was a student of mine who came into my office and wanted to do her undergraduate thesis on humans. And I was like, I don't study humans. And she really was like, well, I want to do that. And so she really motivated the study. So Jamie made the initial contact with the Cleveland Clinic, which is awesome. And they were on board right away, which surprised me, to do a collaboration where they would teach us how to do ejaculate analysis and we would do the studies down at the College of Worcester. And so Jamie ended up not working on that project and doing a great project in fruit flies, but there was another student, Paul Joseph, who took over that project and did this great study on whether human males adjust the composition of their ejaculate and what the implications of that are for human infertility. He worked with college undergraduates and had men come in every other day for 7 trials. In the first 6 trials, they watched sexually explicit clips with one man and one woman, the same man and woman each time, and they produced an ejaculate, and Paul would measure all these different parameters about the ejaculate. And then on the 7th time, they came in and watched a clip that was the same man, but a different woman. So it was a novel female effect. And what Paul found, and the results we were able to publish, was that with this novel female, the males produced a larger ejaculate, ejaculated more quickly, and had a higher number of motile sperm in their ejaculate. After Paul's study, we had so many questions because we had evidence that males were adjusting their ejaculate. And then another student came to talk to me, Eva Katsopoulos, and she said, I want to work on the human study. I was like, that's fantastic. We have a million questions. And she said, I want to work on females. I was like, oh no, oh gosh, okay.
Or, oh, yay.
Yay. I got to yay, but not immediately. I was like, how do we study female ejaculate?
So why was your initial reaction not yay?
I was really excited about the male side stuff. I had lots of questions that I thought we could follow up on logically. I hadn't really started thinking about the female side stuff in humans, and I didn't have any idea how we would test things about the female ejaculate. Eva just took it by storm, and just figured it all out. And so I knew a lot about cryptic female choice. So that's where we started to see, well, is there any evidence that female orgasm is a way that female humans could choose between sperm? Like I just told you with the insects, right? So it was again motivated by the insect. There's been lots of papers written about female orgasm and why it's evolved, because people say it served as mystery because it's not necessary for fertilization. But most of the people who have written about it, not all of them, have been male researchers. And I just thought, well, that's a little strange. It seems like there should be some more female voices in this.
It's not
It's about
important.
female orgasm, right?
We don't need it.
There's other ones like that. It does. So some people have argued that for this upsuck theory, which is that the contractions that happen in the female reproductive tract during orgasm would help propel the sperm through the female reproductive tract closer to the sites of fertilization.
Like the contractions itself are acting as a suction. So going back now to the studies that you've done on the female orgasm, can you talk a little bit about the process of that and how those studies were set up?
Yeah. And so Eva gets all the credit for figuring this out. I had found some papers about pH. I had been very interested in the pH, so the acidity or basicness of the female reproductive tract and of the male ejaculate. And we came together with these interests and wondered whether female orgasm could change the pH of the female reproductive tract. And the rationale for that is that the secretions in the female reproductive tract are really acidic. And they're so acidic that sperm should be dead on arrival. Even with the millions and millions of sperm that are transferred in each ejaculate, literally none of them should make it at the pH that the cervicovaginal fluid is at. But the seminal fluid is really basic. And when you look into some of the literature, it basically implies that the seminal fluid is basic pH is overwhelming the acidic pH of the female reproductive tract. And it sort of feeds into a lot of the sexist language that you see throughout animal behavior literature of like males in control and males being active and females being passive and coy. And so I just wondered if that really could be true. Like it's women's body, and like, is this little amount of ejaculate really overwhelming the pH, or is there something more active on the female side going on? So we looked into the literature, and there was this one study that's been cited a number of times that the pH of the female reproductive tract became more basic after orgasm, so more sperm friendly after orgasm. And I was like, well, if
Oh,
that's true, then it could be a form of cryptic female choice, right? If you have an orgasm,
interesting.
that you would be favoring the sperm of the male that gave you the orgasm, basically, right?
Then you make it a more favorable environment for his
For the
sperm.
sperm, right? That was interesting, but it had been done on like 8 study subjects. And so it wasn't a big sample size. It was about 50 years old, the study, and it was very invasive with like these pH probes up in the female reproductive tract and stuff. So Eva decided to do it in a different way. And she found a study that had collected cervicovaginal fluid from women using menstrual cups. And I was like, well, that's brilliant. And so she got a whole bunch of menstrual cups, wrote out instructions for how people should use it. She tried to get the people in the study to track their ovulatory cycle to see if there were differences in how the pH changed during orgasm, depending on where you were in your reproductive cycle and your ovulatory cycle. But the participants weren't great at accurately following their ovulation cycle. So that didn't work out. Plus, everyone in the study had to be willing to masturbate, use this cup, and also not on hormonal birth control. So it was quite a challenge to find people to be in the
Yeah.
study, just because a lot of college women who are sexually active are also on hormonal birth control. But she did find them, and she did find this trend for just like the previous study had shown, for the pH of the cervicovaginal fluid to become more basic after orgasm. So I'm still looking for a student to follow up on that study. I think there's a lot of cool things to do because we didn't test like the survival of sperm in pre-orgasm versus post-orgasm fluid. That would be amazing to do.
I have so many questions.
Okay.
How many students has it been now since Jamie had that
Since
initial
Jamie,
question?
there's been about 12 students who have done their theses.
That's amazing.
Yeah, it's been really interesting. Some of the students were really interested in stress and the effects of acute stress on ejaculate parameters. So they came up with ways to ethically stress students out, which was really hard. It was kind of very stressful for the students, but I can tell you about that if you want. And then looked at their hormone levels and also their ejaculate parameters. One of my favorites was a team who worked together recently, Marissa Camacho and Kylie Davis, who both studied the relationship between different predictor variables and ejaculate parameters. So Kylie looked at bisphenol A, which is a component of hard plastics and is thought to be an endocrine disrupting chemical. And so she thought maybe exposure to bisphenol A would have correlations with sperm parameters or ejaculate parameters. And then Marissa worked on the relationship between pornography use during sort of early years of your life and later ejaculate parameters. So these were, college age men who were in the study. And she asked, when did you first start using porn? When were you first exposed to it? When did you start using it regularly? What's the sort of degree of porn? Like, is it sort of, I don't know the names for it, but like, does it involve violence or sadomasochism or things like that or not? And a few other questions. And she found a pretty strong correlation with that. The earlier that you've been exposed to porn, the fewer sperm in your ejaculate. So age at first viewing of pornography and motile sperm concentration were significant significantly positively correlated with one another. So that meant that the older you were when you were first exposed to porn, the more sperm you had in your ejaculate, more motile sperm. We don't know. Again, it's a
Crazy.
correlation, not causation, right? And so
Right.
it could be that those men who were exposed as boys earlier on just need like a more extreme form of porn to produce more ejaculate, and that this sort of mid-level that we used for the study didn't stimulate them enough, or it could be that it's really having a physiological effect on their ability to produce sperm.
So, what questions, now that you've had, 12 groups of students that have kind of built on this idea, what questions keep you up at night? What do you want to explore next?
Yeah. So I'd like to follow up on two of the studies. So one would be Marissa's on the relationship between pornography habits and sperm parameters. And is there any causality there? I'm not sure how we would do that, but I want to look into that more. I'd like to follow up on Eva's study and see if the changes in the female cervicovaginal fluid pH affect fertility of sperm. But I also want to start doing some of these studies with non-heterosexual participants as well, and see, are we seeing sort of similar processes with other groups of people? Because at least in the male studies, they all had to be self-identified heterosexual males for these studies. And I think that we could learn a lot from looking at non-heterosexual communities for this.
That's fascinating. I was thinking too, while you were saying that, I know men and women can use vibrators, but does that affect the female pH at all? Because some women prefer them.
Yes. Yeah.
So many questions. I have so
So
many
many
questions.
Yeah.
So, Laura, any last remarks or takeaways that you want to leave for the audience?
Yes, thanks for asking. What I'd like the audience to take away from this whole first season of I Came for Science is that we're all scientists and science needs all of us. So you might not think you're a scientist, you might not have been trained in science or done well in science, but we're all out in the world making observations, coming up with hypotheses, testing our research questions, whether we think we're doing it or not. It's happening all the time. And we really need people with different lived experiences to ask the different questions that nobody has thought of before. And I feel like that's been part of my career, like switching from one field to another, switching from anthropology and math to biology. I asked new questions, switching from primatology to entomology, switching from behavioral ecology to molecular biology, switching between species from primates to insects and then back to humans. And people have appreciated it that I've asked questions that no one's asked before, which is amazing to me. Like, how could little old me, be asking these questions? And yeah, I think it's just being willing to bring your lived experience to your science. And I hope you all will do that too.
Oh, I love that. Yeah, we're all experts of our own lives. So each one of us has a question or something that we've observed that no one else has. So
Yeah.
it's a great reminder.
Yeah. And I think it's an exciting time in science because scientists are starting to acknowledge that scientific knowledge is not owned by academic scientists, right? Like there's a lot of people doing science out in the world, and especially a lot of indigenous communities, people from indigenous communities who have been doing science for thousands of years, really. And, like appreciating that knowledge that's coming from all different sources is really important. So you're out there, you're a scientist. Let's hear from you.
Thanks, Laura.
Thank you, Nat. And thank you all for listening to our first season of I Came for Science. This was the last episode of the season, so I hope you'll join us again in the fall when we launch season 2. And thank you also to our production team, Isabel Espinoza, Sam Carmel, and Baltazar Beretta.